Thanks for coming back and listening to OMG Omics yet again. I hope you'll enjoy this episode, which touches on one of the lesser known omics, glycomics. Hi there, Nicki, and I want to welcome you to OMG Omics as you're into our 2024 season. I was hoping that you would start by telling us about your career and what has allowed you to get to the place of being a distinguished professor.
Hi, Kate. How are you? Well, I guess when I give these talks, usually I start by saying I'm an accidental professor. There was no plan in my childhood that, or even belief. I would laugh at you if you said I was going to be a distinguished professor at any point in my career. It just happened, and I guess it happened. I just followed what I like doing. So, and I took up opportunities. So again, I did biochemistry and maths as an undergraduate. Um, I got a job then. I was the first in my family to go to university. So I thought, well, I've done a degree, I'll get a job now. I got a job in the university, and I was in the lab setting up classes, etc. And I looked at the people who'd done their degree with me, who were doing a PhD. And after a year of working, I thought, I'm as good as they are. Why don't I do a PhD? So I applied to do a PhD, and I did that at Sydney University, and I did that on yeast and lipids in the membrane. So that started my career, I guess, in biochemistry. I then happened to have produced three children. So I have three children. And during that time, which I think probably totaled 10 years, I worked part time in various jobs. So across various biochemistries, people would employ me on their grants. The grants would run out, then somebody else would employ me for another 3 years, and then I'd teach at the technical college. And then I worked in a company doing QC, and so I did all sorts of things while I had the children growing up. And I remember thinking, probably at age 35, that I would be the oldest research assistant in history. Because I didn't have any one thing that I was doing. Then I came to Macquarie University for a job in lipid polysaccharides, which you'd be aware of. And from there followed I was on a fellowship. I then got another fellowship, and then I dropped out of the university, and we formed a company called Proteome Systems. It was at the beginning of proteomics. It was all starting, and my boss Keith Williams said, "Look, we just can't get enough money in the university system to make this happen. So how do you feel about joining me in forming a company?" So five of us jumped ship and formed a company. The company, I have to say, lasted 9 years without listing. We did all sorts of things in proteomics. We did discovery, we did service, we did bioinformatics, we did robotization. But when we listed, were doing too much. Basically, our revenue didn't equal our costs in any way. So it crashed and burned. A friend of mine back at Macquarie University said, "Are you ready to come back into the university?" So I said, "Sure." And because I'd been paid this much in the company, they had to pay me at a professor level at the university. So in many ways, I skipped all those things that currently postdocs are told they have to do, and came back in at the professor level. So I guess that's a quick summary of my long career.
So I know that you're now at Macquarie University, but don't you have other affiliations? And how do you take the different things that you do and balance them all out?
With difficulty. They say that being a mother makes you able to multitask, and I think that's a very good example. Yes, I have a part-time affiliation at the Institute for Glycomics at Griffith University in Queensland. I am chair of the Human Proteome Project Biology and Disease um, part. And I'm in the Human Glycoproteomics Initiative, also of HUPO. So I have an active role in HUPO. What else do I do? I don't know. I talk to Brooker a lot. I talk to mass spectrometry companies a lot. Oh, yes, I'm also the academic lead of the Australian Proteome Analysis Facility, which I was part of setting up probably now 25 years ago. It was one of the first facilities that provided proteomics services. I then left to do what I was doing because my boss was the person who set that up as well. And now when I came back to the university, it went full circle, and they asked me if I would be the academic lead of the facility now. So that takes up some of my time as well. So I'm not sure what else I do, but yes, that'll do.
It sounds like you certainly keep plenty busy.
I'm pretty busy. Yeah.
Yeah. You said something else a moment ago that I find interesting. You are a first generation college student, and you must still encounter a fair number of those. What's changed, and what's the same that you notice from when you did that to now?
Hmm. I don't know. The general feeling is that a lot has changed, but I'm not convinced that a lot has changed. Um, I think the pressure now on postgraduate students is much higher. That what they should do with their life is get a fellowship, and then they should get a position in a university, and then they should go from level down here up to level up there, and they should publish, and they should teach, and they should do all these things, which is one route. I agree, and that wasn't pushed at me at the time. I didn't have in my head that this is what I had to do if I did a PhD. Whereas I think the kids now are being told that this is what you have to do. You've got a PhD, so you've got to do this. In my experience, there are other routes. There are other things which give you satisfaction. There are other ways to make a living. There are other things to do. So I think that's probably changed, and people need to be told that there are other ways that you can use your PhD to good value. I think, Kate, you would be one of those. so, um, so I think that's changed. Probably the other thing which has changed, but not fast enough, is the opportunities for women and men. I think I could go on about that. That even in my span, the next generation, it still is struggling to make that role equal. And I don't mean equal in money or just equal opportunity to the men actually pick up a bit of the slack on this. I could go into a women's rant, but I won't. But basically, my advice is less to the women and more to the men because you actually enjoy having children and spending time with them. So if it makes your career flatten for a little while, so be it. It will kick off again if you're any good. So that's, I guess, my main mantra at the moment.
I appreciate those 2 cents, as we would say in America.
Yes.
So maybe something else along this line. I think your career has been intentionally structured to continue to support people in science after you step away from the main stage. Can you tell me a little bit about how you put that together?
Yeah, it started in my head really when I was in the middle of my career, and I visited a professor friend of mine, and he was retiring, and his lab was being closed because someone else was going to be appointed, and they would be doing something completely different, and his work would not continue. And I felt at the time then that that was such a waste of everything that he'd done, everything he'd achieved, and the field that it had lost that continuity. So I guess in the back of my mind, and it was when you say intentionally, probably a little bit in my subconscious rather than my goals, was to make sure that the field continued. after, as you have put it, after I stepped down from the stage. So I have probably now several postdocs who are keeping the ball rolling. In the Institute of Glycomics, he has just been made a professor of glycomics, Daniel Coleridge. He was a postdoc of mine some years ago. Here, Associate Professor Morton Anderson, who also came to me to do a postdoc, has now set up his own research group, and it's doing very well. And now at the moment, I have two of my ex-students who have come now to work with me again, and they are building up their careers as well, and they're very firmly entrenched in glycomics analysis. So I sort of that's Edward Moe, and Catherine Wong Kish, and both of those are dedicated now. So I feel that I, when I step down, there is a very good succession line that will could keep the science going. Yeah.
So maybe let's turn to the science, and maybe you can tell me a little bit about how you've seen proteomics, you've seen glycomics. Tell me about the interplay between these two fields and help the viewers understand that they're not necessarily distinct.
They are definitely not distinct. Proteomics started, as you probably know, you identify proteins. But what you do is you throw away all the peptides that are translated or are modified in some way in the mass spectrometer. So the software only looks for the ones you're looking for. So you see what you see and you identify proteins. And proteomics became automated largely with software that identified lots of proteins in lists. And all the rest was ignored. And in function, if you look at those 20,000 genes in the human genome, you know, 50% have been thought to be modified or glycosylated, and probably they're all modified in some way. So you've got millions. In fact, it's power orders of magnitude greater of actual proteins that are operating out there. And my interest coming through the lipopolysaccharide was carbohydrates and and that's how I got into uh, glycoproteomics from proteomics. And when I realized that, just 50% of the proteins had a sugar, not one sugar, many sugars, and the heterogeneity was enormous. And my belief is that biology does not operate making things if they're not used. So there must be some reason that these proteins are modified in this way. They must be doing things. And the difficulty between phosphoproteomics and glycoproteomics is that phosphate is only one mass. It's on a couple of amino acids. It has really similar function in signaling, etc. Whereas we're finding that glycans are involved in all sorts of functions, things you can't predict. Suddenly are affected by the glycosylation of the protein. So the field is, in that sense, linking structure to function is huge. So we can now start to look at glycans more easily, but we still haven't completely got it together. And now I have to say, for the non-proteomics uh, slant on this, there are more than just glycoproteins. There are glycoconjugates would be a better word. So there's glycolipids, there's proteoglycans. You go into the bacteria and you think, oh my God, there's lipopolysaccharides, peptidoglycans. They're everywhere, let alone polysaccharides by themselves. So it's a very big field which has not, I think, been exploited enough for its functions. And in the same way as proteomics did not flourish until the tools were there and the software to understand those results was there, proteomics did not function. And the old story I had is you have the PhD used to be one protein, one function. You take your whole PhD looking at what this protein did. Proteomics opened up to say, look at all these proteins, look how they're changing. So it only happened when the tools were there. And glyco is getting there. It's not there yet, but it's certainly getting there.
So, what do you think? You hinted at this, but maybe you can expand a little bit more. What's the current biggest need in glycomics to determine activity and biological impacts of some glyco compounds?
Glycans are so structurally diverse, so there can be different linkages which determine different activities. So I'll give you the obvious example at the moment, not at the moment, has been, is influenza virus. It actually can tell the difference between a human and a chicken by the linkage of sialic acid, one monosaccharide, to its surface sugars. Not even, doesn't care about the proteins, just which sugars are displayed in the throat, in the nose, in the mouth. And the influenza virus will see a different linkage in humans, and then the virus, the influenza virus that infects chickens, for example. And that is the main reason why we have not had the transfer of the avian flu into humans, is because the avian flu virus doesn't recognize the linkage which we predominantly have of sialic acid in our respiratory tract. So that's one example. Um, so I guess when you think what is the holy grail, or what we need to do to link structure and function, is just to look at the structure in detail and then see what it does when you change that structure. And what this example you're talking about is a peptide that my student has just submitted his thesis, so I'm very familiar with that one at the moment. So it's a short peptide, it's got an immune function, and it's got one N-link site and some O-link possible sites. It took him some time to characterize the heterogeneity of that glycopeptide because on these sites they had numerous structures. Then when you separate that, so they're trying to make a recombinant peptide, glycopeptide, they put it in a recombinant system and you produce these glycopeptides, and then they put it through an anion exchange column, but only 1.5 fractions actually had activity. So less than 1% of the recombinant glycopeptide actually had activity. Now, Nick worked on this for some time, and a student before him, worked on that as well. And the people that discovered this had worked on it for years before that. So it's a very long story about what confers activity to this peptide. And it's obviously glycosylation because that's what's on it. But which one? What combination of that can be? And so the characterization had to be really detailed, like what the linkages are, what the sequence is, how much is there. And at the end of the day, he came up with there's this structure on the N link, there's this structure on the O link. And then you go, yeah, but what does that mean? We then passed it to Lisa Fadder, who's a structural modeling scientist um, who loves glyco. We finally got one. And she modeled this and found that the N-link sugar had one effect, one function, and the O-link sugar had another function. And without the structures on those two sites being what they are, you just got no activity. So that's the challenges we have going forward. So there's lots of work to do, lots of jobs out there. I can honestly say that I don't think anyone who has passed through as a postgraduate in my lab has not got a job. They all have a job. They are all in demand. So I think that's the future, really.
So you have to have a robust toolbox to be able to do all of
Yeah.
these analyses. Tell me
Yeah.
about your first introduction to mass spectrometry and how you've stayed with that as one of your key methods.
Yeah, absolutely. It was at Macquarie University in the School of Chemistry, and there was one mass spectrometer and one operator of that mass spectrometer. And when I went into the lab, it was always in pieces. He always had the source out or the tubes. It was always in a mess on the floor. And it was really, I think, proteomics, the rise of proteomics, which made the mass spec companies pay attention. and see a market out there because people wanted to be able to fragment proteins. And so then the developments, well, Moldy was already there, but electrospray came on in all its many guises, and so it's gone on. So I think that proteomics kicked that off for the companies. I think the companies that are out there now in mass spectrometry probably had a ramp up at that stage from just doing small molecules into doing proteins. So it's been quite a revolution there. And now, of course, mass spectrometry is our main tool for the glyco field as well. Um, but you have to operate it a little differently. So, and I think that's what stopped people for a while because I would talk to people at conferences and I'd say, why aren't you doing glycomic analysis? And they say, oh, it's too hard. Right? But it's not too hard. It's just you need to know how to do it. And the other thing I can say about being too hard is both DNA and proteins are linear, structure aside, but they're linear sequences. Sugars are not linear, they are branched and they have different linkages. So there's no one enzyme to hydrolyze it, there's no one anything. So it is complex, but it is able to be done now, not only by mass spectrometry, but you need the LC in front of it as well. So it is definitely best done, I believe, with a LC-MS system.
Nicki, it's been really great chatting with you and hearing about your story. I'm wondering if you can tell me a little bit more about some of the people, maybe what your research group is like, beyond just some of the names that you've dropped.
Okay. At the moment, you mean the research group I'm running at
Yeah,
the moment? Is
yeah.
that? Yeah. Okay. I think last count there were probably 12 of them at different levels. So I don't teach any more undergraduates. I used to, but I don't anymore. I'm just on a research group and how I'm running it, which suits me beautifully because I don't actually press the buttons anymore on the mass spectrometers. That's I've left that now. But I have different levels of scientists. So I have now 4 postdoctoral fellows who are mass spec friendly, and actually I just got one more, so that's 5. And they sort of, so I've got a structure now where I've got people who know what they're doing in the mass spectrometry field, know glycosylation, they're all glyco fanatics, so that's all there. And then there's the PhD students that sit under that, and then there's the Masters of Research students that sit under that. So the whole group is a young group of really cool people who like doing what they're doing. We just went down to a conference as a whole group and hired a house and at the Lawn Proteomics conference, and we hired a house and hung out together. And so I think that's great for bonding, for enjoying science, for sharing stories, etc. As well. So that would be my group at this moment, and I'm really enjoying it. So people say, "When are you going to retire, Nicki?" And I go, "Well, when I stop enjoying it."
So, what else do you do? Maybe as a last question, what else do you do to ensure you have the balance so you do still enjoy what you do?
Yeah, balance is important. And I can honestly say that family is my balance. So I have 3 children and 2 grandchildren, and I went part time, so I'm a total of zero point 0.7 70% employed at the moment, and I spend one day a week looking after my grandchildren, so that my daughter can, who's also got a PhD, can continue to work and continue her career, albeit again part time. Then so I'm involved probably in that in my spare time. Yeah.
Well, thank you so much for sharing a part of your story with us, and I look forward to seeing what you continue to do in your career.
Okay. Thank you, Kate. Thanks very
Yep.
much.
I hope you found Nicki's story compelling and her ability to balance things across her life and career a model for some of you out there who are trying to figure out what your path may be. Thanks for tuning in, and we'll see you next time.